All right, so here we are, the last part of this chapter 3. We're going to start talking about the nucleus. We're just going to go over some very basic kind of things, so this should be fairly quick. All right. So remember, you have 46 DNA molecules or chromosomes in your nucleus. Remember, you get half of those from mom, half of those from dad. Right? So 23 from mom, 23 from dad. Genes are found on chromosomes. Remember, a gene, all that is simply a recipe for a protein. That's what it is. It's a recipe for a protein. That's what your gene is. Okay, so depending on what that protein is, it might be a combination of genes working together to make it, or it's a single gene sort of phenotype, if you will. And I will hide my image so you can kind of see the rest of this picture here. When your cells are not dividing, the chromosomes or this genetic information is in the form called chromatin. So basically, the chromatin is easy to access version of simply your chromosome. Okay, so I kind of think of it like this: when your cells are not dividing, it's sort of like you living in your house, right? You may have all your cookbooks or all your books are out on shelves, so they're easy to access, right? But let's say you buy a new place, and so what you're going to end up having to do is you need, all right, I'm just going to spend half your time here and half your time there. Well, what's going to end up happening is now you need 2 of everything. So you're going to replicate that, and then what's going to end up happening is you're like, all right, now I got to box up this half that I just made, so it's easier to move. So when you're boxing things up, that's like your easy to access chromatin fibers getting kind of bunched up, boxed up, packaged. So it's an easier to move in the form of a chromosome, okay, or a chromatid, I guess, over to the next cell. Or, well, yes, over to the next cell or to your new house or apartment or whatever it might be. Okay. All right. So again, what the DNA has is simply the instructions for protein synthesis. That is it. And so somehow those ribosomes need to know how to make them, so they are simply following the recipe, the instructions found in the DNA. That's why it's so important that your DNA remains undamaged. If you end up having some sort of damage to it, then what ends up happening is when you're copying that over, you're copying it over wrong. And some mistakes are worse than others, right? Like if you use, I don't know, in a recipe, use sage versus rosemary. All right, it might not be the right flavor, but it's not horrible either. However, other copies, other mistakes can be worse than other, So for example, if you use salt versus sugar in baking, ooh, that's no bueno, right? The taste is going to be completely different and the recipe is ruined. So again, depending on where damage is in your DNA, it can be a bigger problem or a smaller problem. And the reason why is because it drives protein synthesis. So if you got a bad recipe, you're making a bad protein. Again, and pretty much vast majority of you is protein. There's very few things you can be made of, right? You can be made of protein, lipids, and sugars. There's some minerals in there and some other things, but those are the main categories. So what you're going to see over and over again, a good chunk of you is protein. So to make protein, you have to go through transcription and translation. Now again, we're just going to talk about this at the very sort of superficial level. Now imagine this. Transcription, so let's say nucleus is sort of like a library, okay, if you will. You know how you have some books on reserve at the library and you can't take them home with you, right? So you want to get a copy of this recipe and you want to do it at home, right? You actually want to make that, I don't know, whatever, dish at home. So you're going to have to take that recipe at the library and you're going to have to copy it down into a form that you can understand, right? So that's basically what transcription is. That's all transcription is. All you're doing is you're taking that recipe at the library and you're turning it into a form that you can understand at home. Okay? So the form in our cells is we take that DNA and we turn into RNA, specifically messenger RNA. So now those ribosomes can read that and they go, oh, okay, you want to make, I don't know, neurotransmitter A, right? And they go, okay, we can do that. So first you transcribe it, you copy it over. And then you can take it out of the nucleus. And then, once you have it, now you translate it. You take it from that mRNA, which is just instructions. It's just that piece of paper with, hey, add in, da-da-da-da-da. And you're actually going to take that information and you're going to make it into something. You're translating it. So you're taking the mRNA and now you're actually going to make protein. Okay? That's all it is. That's it. How simple is that? Yay! So that was that. Cell division, again, not going to go into like major details here. It's simply going to be the process by which cells are going to reproduce themselves. So the two parts to it are nuclear division, and that's what we're talking about when we talk about mitosis versus meiosis. And I know people get those two mixed up a lot. Again, really simple, super simple. Mitosis is basically everything, except when you're making either a sperm cell or an egg cell. Meiosis is if your end product is a sperm cell or it's an egg cell, it's meiosis. So all you do is you're like, well, what did I make? Did I make a skin cell? It's mitosis. Did I make a hair cell? Mitosis. Did I make an eyeball cell? Mitosis. Tooth? Mitosis. Anything. Is it an egg? Yes. Oh, meiosis. Is it a sperm? Yes. Oh, meiosis. That's it. There's a big difference. That's pretty easy too. So people get really confused. I don't know why. Your end product, sperm, egg, meiosis. Everything else. Everything else. Mitosis. Okay? And again, when we talk about mitosis and meiosis, what we're talking about is the division of the nucleus. That's what we're focusing on. But you got to remember, the cell is more than just the nucleus. So the rest of the cell splitting apart is going to be called cytokinesis. So that's sort of the difference there too. We've got the nucleus, mitosis, meiosis, depending on what we're making, and then cytokinesis, just the rest of the cell dividing. And I'll kind of hide that so you can see the rest of that. So here's just a brief review on the two. So on the blue side here on the left, this is mitosis. So remember the different phases. There's prophase, metaphase, anaphase, telophase. I'm going to give you a nice and easy way to remember these in lab. Very briefly, I remember it as p-mat. So like a dog peed on the mat. I don't know, it works for me. So prophase, metaphase, anaphase, telophase. And like I said, I'll give you a nice, easy way to remember what is occurring in each one of those phases in lab, because we need to go through mitosis anyway. Meiosis is exactly the same thing as mitosis, except you do it twice. So that's really it. And then there's only one other thing, and that's right here in this prophase 1. You have what's known as crossing over. So in that crossing over, you can swap genetic information. So that's the only other difference. So, in mitosis, you go through that p-mat once, and it's just done. So in meiosis, you do it twice. And you have this crossing over, which is the swapping of genetic information. Okay, so you can get different egg cells and different sperm cells. If you'd like a review on either one of those two, I've got a couple of YouTube video links here. Feel free to watch them. Don't worry about going into super detail. In these, I'm not going to go into super detail. The only other thing that I want you guys to remember is whereas you have 46 chromosomes that's the end product in mitosis, right? The end product is 43 in meiosis, because again, you need to have it so you have half from mom, half from dad. So 23 versus 46. That's about it. Cancer is simply out of control cell division. Well, not simply. It's a big problem, but that's basically what it is. It's just cells not being able to regulate their division properly. Okay? So they exhibit something called hyperplasia. So the number of cell divisions is increased. Okay? So they're dividing sometimes too rapidly. Okay? We have different terms. So when we say a tumor is benign, that means it has not metastasized. Metastasize simply means spread. It hasn't moved from wherever it is to some other organ. A malignant one, on the other hand, is one that there's like a little piece of that tumor is broken off, and it has moved either in the blood or in your lymphatic fluid to some other body part. Okay? That's one of the reasons why a lot of times when they go in and they take a tumor out of a body part, they'll also take out the lymphatic tissue nearby, because sometimes it gets stuck in there before it can move on to wherever it's going to go. So if they can get it out of there before it moves, they can prevent it from metastasizing, from spreading somewhere. There are many different causes to cancer. Obviously, carcinogens, those are cancer-generating agents. A lot of times chemicals, different materials, different things like vast number of X-rays or other kind of radiation, I guess. Viruses can also cause cancers, and that's one of the things we've learned about in the recent past is viruses can also cause cancers. Oh, there's just like recently, like a few weeks ago, in the news, well, I guess not for you guys a few weeks ago, so it's a few weeks ago now. So this is November of 2015. So just a few weeks ago, they had a case of where a tapeworm actually infected a guy with cancer in his intestines, which was like unheard of until then. So tapeworms can also cause cancer. Okay. So again, cells are going to regulate their division. So what ends up happening is, whether it's radiation or virus or whatever, is going to cause a mutation in those genes that are regulating how fast and how much they divide. Okay. Usually it's not a one-time event unless you're really exposed to like a very large dose in a single sitting, and that does happen in some accidents. But more often than not, they usually develop cancers later on in life. That's usually due to an accumulation of exposure to different mutagens that then finally collect. Your cells can't fight it off. Finally, there's some sort of error. You replaced the sugar with the salt, and now you have a problem. There's different types of cancer, so I'm just going to present to you a couple of the more, prevalent types. So a carcinoma, and there's many different subcategories of these. So these are sort of the big umbrella terms. So carcinoma is from epithelial cells, and epithelial cells will be cells that are lining things like your skin, the inside of your mouth, the inside of your organs, so inside your stomach, your esophagus, intestines, those kinds of things. Those are carcinomas. Melanomas are due to melanocytes. So melanocytes in your skin are what are giving you your sort of either tanned appearance or freckles or something like that. So sometimes these melanocytes, they accumulate a lot of this damage. They're one of the things that help protect you from like sunburns. So they accumulate a lot of radiation damage from the sun, UV rays, and then eventually they start dividing rapidly. Sarcomas can either arise from muscle cells or other connective tissues, so things like cartilage, bone, what have you. Those are sarcomas. And leukemia is cancer blood-forming organs, so the main blood-forming organs in an adult are your bones. So usually when someone has leukemia, it's a bone disease. You've probably heard of it, like you'll hear people getting bone marrow transplants when they have leukemia, because they have to replace that part of the bone that's actually forming new red and white blood cells. Lymphoma is simply cancer of the lymphatic tissue. One of the things too with cancers is because you have rapidly dividing cells, they're going to need a lot of nutrients, right? So they can create all these extra organelles and stuff. So they're going to need a very good blood supply because that's where nutrients are going to come from, right, is the blood. So these cancer cells, or these tumors I should say, can trigger angiogenesis. Angiogenesis means the growth of new blood vessels. So some of the drugs these days, they actually target new blood vessel growth because the idea is if you prevent the cancer cells from triggering new blood vessels, you'll essentially starve the tumor. Cells can't divide if they're starved. There's nothing for them. There's no materials for them to use to build more cells, and eventually the tumor will simply die. Again, and we already spoke about metastasis, right? So that's the end of chapter 3. So hopefully you've taken all your quizzes. And if you have any questions, feel free to email me or ask me in lab. So on to chapter 4. So see you then.